Hi, I'm Zhuhao Wu from Mount Sinai in New York. First, I want to thank the organizers for this great meeting and this inspiring symposium. Following the last exciting talk, I will discuss more details for tissue clearing techniques on human brain samples. Together with our collaborators that you will hear more today, we aim to leverage on tissue clearing and light sheet imaging to map out every single cells in the entire human brain. For this audience, I don't need to emphasize how important yet challenging it is to profile the entire human brain with cellular details, covering its many anatomical compartments and the enormous cell type diversity and connectivities within. We have seen many exciting progress to better comprehend the entire mouse brain, but the human brain is 1,000 times, to be more accurate, over 2,000 times larger than the mouse brain, with many other challenges that I'll touch upon later. Even the relatively small mouse brain is not that easy to study in its entirety. We have been developing and applying tissue clearing techniques to make it fully transparent, as you can see here, so we can acquire fine details inside using fast light sheet imaging without the need of physical sectioning and laboratory reconstruction. If we can scale this fast approach to the entire human brain, then we can hope to finish it within a reasonable amount of time. Another challenge is that unlike mouse brain, with many available genetic tools and reporters, we need to rely on labeling endogenous markers for the human brain samples. Here, I want to cover the general procedures of our tissue clearing. It sounds like a magic to make a biological tissue fully transparent, but it is indeed following basic physical and chemical principles that I'll discuss. When light travels through a biological tissue, it will get deflected or affected by the cell membranes. It's not just a plasma membrane, but also the extensive membranous structures inside the cell, like the Golgi network and various organelles. Therefore, most light will get scattered off after just a few cell layers. In order to make a tissue fully transparent, we first need to remove the membrane lipids. After complete delineation, a fixed tissue essentially becomes a nearly homogeneous protein scaffold. That if we can match its refracting index with a high refractiveness buffer, then it becomes essentially fully transparent, or now the light can travel through it without any problem. Over the years, we have been optimizing our procedure following these general principles that I just mentioned. For delineation, we have been engineering our lipid extraction buffer that can solubilize both the hydrophilic head as well as the hydrophobic tail of the polarized lipid that are enriched in biological tissue, while preserving the protein scaffold in its native form. For refracting index matching, it is indeed difficult to measure a brain's refracting index directly. However, we can extrapolate 100% hydrated BSA to be about 1.5 to 1, based on its linear relationship of the concentration and its refracting index. Using this number as a starting point, then we can approach that a brain's refracting index is about 1.5 to 9. I do want to point to you that the brain's refracting index may depend on the actual procedure. During our optimization of the procedures, we have been trying to delimitate the sample completely while preserving the protein scaffold in its original form, or therefore the overall refracting index would be fixed to a very narrow range that we can match it really well. With this said, we have been optimizing and developing our tissue clearing protocols through iterations over the years with my extended collaborators. Showing here are some examples done by our original protocol iDISCO that are still being widely used in the field for robust clearing and labeling various tissues covering brain and other organs to profile the fine anatomical details inside. I do want to point out that iDISCO do shrink the tissue a little bit, and for the brain, it shrinks the cortical region a little bit more than the subcortical region. This issue is addressed in the second version of our protocol, iDISCO+. A brain image with iDISCO+ is well preserved in its morphology so that the HuMA imaging can be registered back accurately to its original MRI scan or to the standard atlas. This enables us to develop automatic pipeline for brain alignment registration and quantitative analysis, which is critical for handling large datasets generated by the HuMA imaging. We have further enhanced the lipid extraction procedure in our protocol so that it can not only clear brain, but also fat tissue, which is the most lipid-enriched organ in our body. Hence, this protocol was called AdipoClear. AdipoClear can directly extract triacylglyceride, the major lipid depot in the fat. Brain also has a trace amount of triacylglyceride as a normal metabolic byproduct. It could be rapidly accumulated during injury and aging. Therefore, if you use AdipoClear to clear a brain, you can also get more superior clearing performance. Together with other improvements, AdipoClear preserves the tissue indigeneity very well, so that now majority of the IHC validated antibody is compatible with our hormone procedure. Because our clearing is following on general principles that are not only well conserved across different organs, but also across different species. Therefore, we can combine all our recent advancement in tissue clearing and directly apply for the human brains. Also, our procedure is based on chemical engineering. So, in another word, all that we need to do is find a bigger jar with a human brain slab into it and run through the same chemical procedures, and then you'll become transparent like what you can see here. So this is a medium-sized human brain slab from 89 years old donor after 4 months formalin fixation, and it still worked quite well. We are currently optimizing and adapting the human brain clearing procedure for a whole human brain imaging pipeline with cellular resolution. Of course, this is being done in collaboration with a team of great scientists. Our Human Brain Bank Director, John Query, will help to screen and procure human brain samples in the best conditions that we could. Then Alan Zephyr will take the best prepared sample for MRI imaging. MRI imaging will be utilized for 3 major purposes. With the help from Bradley Delman for radiology reading, we'll understand the pathological status of the sample. Then the MRI imaging will serve to incorporate the MRI-based atlas to the sample that we're going to process. Finally, MRI imaging template will serve to incorporate the later high-resolution human brain slab imaging to the original brain space. Putting all these together, we can map fine anatomical and pathological details in the brain sample that we're going to process with help from Patrick Hoff. I'm not going to talk about the imaging, which will be done by our great collaborator Elizabeth Hillman at Columbia, leveraging on her super fast light sheet imaging system, and with her extended collaborators at Columbia for initial data processing. The data will be deposited at BIO, which is led by Alex Roblowski's team. What I will be focused on today is talking about the sample preparation. We'll take the 5 ml thick brain samples, turn it into fully transparent and labeled format to feed into the imaging pipeline. We have optimized our hormone labeling procedure for human brain samples. As you can see on the top, here is a Norse labeling, and you can appreciate the sparse cell body distribution over centimeters in the human brain block. And bottom left here is NPY labeling for another distinct neuronal subclass. Not only you can appreciate the cell body distribution, you can also see their extensive neural projection in both gray matter and white matters. In the bottom right here, green labeling every single nuclei, while the magenta is the arteries coursing through. As you can see that the labeling is even and complete over centimeters in the entire human brain block. We have identified dozens of markers that could label diverse structures in human brain samples, while we are planning to screen many more monoclonal antibodies to cover over 500 neural targets for compatibility. Such kind of data set is not currently available in the field, which we believe is a major limitation for people to adapt for human brain hormone to IHC. We'll share our result online. We focus on monoclonal antibody. Of course, it's because it's reproducibility, but also because it's scalability given their renewable reagent, which is critical for providing large samples like human brain, among other factors that I'm not going to discuss today. Here is our antibody screening pipeline. We'll take the publicly available monoclonal antibody resource that everyone can have access later and screen it over mouse and human brain samples, utilizing our optimized 6-section IHC protocol. We use our 6-section IHC protocol because it's a nice bridge between the standard histology to hormone application. So our screening result will not only have a good predictive value for the later hormone success rate, but also it can serve as a good reference for broad IHC applications. We choose the top candidate from the validated monoclonal antibodies and do a hormone processing on mouse brains as a proof of principle. Both our extensive 2D categorization as well as 3D profiling on selective targets will be released through our website portal. From this data, we'll try to design the best strategy to label human brains in whole mount. This would project to generate a much larger database, as Elizabeth will discuss later today. Here are some of the data you'll be able to find from our web portal currently, covering early screening result of monoclonal antibody on mouse brain samples. As we are ramping up our project currently, we hope to upload more data covering both mouse and humans soon. Eventually, from our web portal, you'll be able to find both curated monoclonal antibody for diverse cell types and neuronal structures with 2D or 3D reference dataset. More importantly, you'll be able to find our detailed tissue IHC protocol. We believe detailed protocol is critical for users to successfully implement the resource that we're going to generate. Here are some of the recent results that we screened cell type markers on human brain samples. Along this process, we are also optimizing the human brain IHC performance to see what will be the best optimal result for each of the given marker. As you can see here, I'm using relatively short-fixed human brain samples. What I have seen over a battery of different markers that human brain samples fixed for 3-day or 4-degree generally give me the best performance. After knowing what we should expect, we have also incorporated a Hohmann antigen retrieval procedure to work with long-fixed human brain bank tissues. In general, longer fixation will start to decrease the sensitivity of most of the monoclonal antibodies. Some are more sensitive than the others. With Hohmann antigen retrieval, we are able to restore many of the markers to its expected performance compared to the short-fixed standard. We hope this effort will help us to achieve quantitative analysis of human brain sample with IHC, with understanding of various conditions that might influence the IHC performance outcome. Eventually, we hope to utilize the battery of markers we're establishing here to not only understand how the cell type composition is across different brain regions, but also understand how they may dynamically change during development, aging, and disease. We hope that these new tools and resources that we're building for cellular and molecular profiling of entire human brains will enable new possibilities for us to understand how the brain structures is being composed of, how brain functions, and how these functions may get disrupted in various diseases. I'll stop here, and I want to acknowledge the members of my group for the large amount of work that they have been doing to push forward the development of this exciting technical platform. We hope that the tools and resources that we're currently building will help facilitate the field for molecular and cellular mapping across the entire human brain. I also want to acknowledge my great collaborators, especially the Holy School. It has been so exciting to work with all of you. And my funding support, this project is largely supported by the Brain Initiative funding. Finally, I want to thank for all of you listening to my talk. I hope next time we'll get to meet in person and have more extensive discussions along this project. Hope you enjoy the rest of the exciting talks and have a nice day. Bye.